Let's talk about 3.3 meiosis. Here are the understandings, applications, and skills for this topic. So let's talk about understanding 1, meiosis in outline. So cell division can either occur via mitosis or meiosis. And the differentiating factor here is that mitosis produces 2 diploid nuclei, so 2N, which is diploid, and meiosis produces 4 haploid daughter nuclei, or just N. And we get this, 4 of them in meiosis, and in mitosis we get 2 diploid nuclei. So meiosis cell division produces 4 haploid daughter cells, which each have 1 of each type of chromosome. So they have half the number of chromosomes of the parent cell. So let's say that the parent cell has 46 chromosomes. Well, when we do meiosis cell division, we're going to get the end product is going to be 4 haploid daughter cells, and they're each going to have half of the original. So each is going to have 23 chromosomes. And then the second point is the nucleus divides twice. So we have meiosis 1 and meiosis 2. And we can see that down here. So meiosis 1 first, and then we have meiosis 2. So in the first division, we only have 2 daughter nuclei. And then with meiosis 2, we end up with 4. Understanding 2, meiosis and sexual life cycles. So reproduction can either be sexual or asexual. Let's talk about sexual reproduction. This type of reproduction produces genetically unidentical offspring. So this means the chromosomes are going to be different. Asexual reproduction, we get genetically identical offspring. So the chromosomes are going to be the same. So the words in bold are the differentiating factors between sexual and asexual reproduction. And we can see this right here. So unicellular organisms or potatoes or plants, they have asexual reproduction. And us humans, we have sexual reproduction. So in eukaryotes, sexual reproduction involves fertilization. So what is fertilization? Fertilization is the union of gametes from 2 different parents. So the amount of chromosomes is going to double. So if I get my pen again, so let's say this egg cell, we know it's a gamete, so it's going to have 23 chromosomes, and so is the sperm cell. And when they fertilize, the union of the gametes from 2 different parents doubles the amount of chromosomes. So this zygote is going to have 46. The chromosome number of species would double every generation, if not halved. So let's say we have 46 chromosomes in a species, and the next generation, it's going to be 92. And to prevent this from happening, we have a stage called meiosis, which divides a cell to be 23. And then when we have two gametes, a sperm cell and an egg cell, they come together to give us 46 again. So the original chromosome number is maintained, so the species have the same chromosome numbers as the generation increases. So meiosis can occur at any time during the sexual life cycle, and in animals this occurs during gamete creation. The fusion of gametes and sexual life cycles cannot occur without meiosis, so meiosis is quite important. Replication of DNA before meiosis, understanding 3. So we know from topic 1.6 that DNA replicates in interphase prior to cell division. So this cell division is meiosis 1. And in interphase, which we can see right here, we know that DNA replicates in this phase right here called S phase, or DNA synthesis stage. So we initially are going to have 46 chromatids, and it's going to replicate to 92 chromatids. And they are going to arrange themselves in homologous pairs. So these 92 chromatids arrange themselves in homologous pairs to form 46 chromosomes. How do we get 46 chromosomes? Well, we just divide 92 by 2 because they're going to be arranged in pairs. So we're going to end up with 46. And we can just see that here. Bivalence, formation, and crossing over, understanding 4. So in meiosis 1, which is the first division, homologous chromosomes pair up, and this is called synapsis. And synapsis forms a bivalence. So if we go here, we can see that this right here is a bivalent. So what is a bivalent? It's essentially a pair of homologous chromosomes. So we have one chromosome right here and another chromosome right here, the blue and red chromosomes. So these are homologous to each other. And when they are joined together, it forms a bivalent. So each chromosome has two sister chromatids. We can see here the red chromosome has one chromatid running down here and another chromatid running down this side. They're connected by the centromere. A bivalent is going to have 4 DNA molecules because they have 2 chromosomes. So we have 1, 2, 3, and 4 DNA molecules. And we can just see that right here as well. So right after synapsis, again, synapsis is just when homologous chromosomes pair up. Right after synapsis, a process called crossing over occurs. So crossing over. And crossing over is when non-sister chromatids are joined together at certain points. And this point is called a chiasmata. So let me just get my pen again. So we can see that this region right here, chiasmata, or in the region of this chiasma, we're going to have crossing over. And what is crossing over? We can see this image right here. So crossing over is occurring. So essentially, the two non-sister chromatids of each chromosome are going to come together in the chiasma region and exchange particular genes. So we can see that this gene has been exchanged. These 2 genes have been exchanged within the 2 non-sister chromatids. It's important to know that this only occurs between the non-sister chromatids. So what do I mean by non-sister chromatids? It means chromatids that are not from the same chromosome. So crossing over occurs at random points, so it can occur anywhere along the chromosomes. And at least one crossover occurs in each bivalent. So if we go back to this bivalent right here, at least one crossover is going to happen. However, many crossovers can occur. And it occurs on the same position on the 2 chromatids. So we can see back here, it occurred on the same position on the 2 chromatids. So the same locus. So there is a mutual exchange of genes. Same alleles of exchanged genes are likely to be different. So chromatids with different combinations of alleles are produced. So this leads to genetic diversity. Random orientation of bivalents, understanding 5. So the attachment of microtubules is not the same in meiosis as it is in mitosis. And these are the simple principles for these. So each chromosome is going to be attached to one pole only. So let's see, if we go back to this bivalent, this red chromosome right here is only going to be attached to one pole. So if I get my pen and I draw the cell right here, and if I have this red chromosome, it's only going to go towards one pole. It's not going to go toward either pole. That is not going to happen. So if we go back here, the second point is the 2 homologous chromosomes in a bivalent are attached to different poles. So, and if I go back to that slide again, the red and blue chromosomes are going to be attached to opposite poles. The pole to which each chromosome is attached depends on which way the pair is facing, and this is something called orientation. Orientation of bivalents is random, so each chromosome has an equal chance of attaching to either pole. The orientation of one bivalent does not affect other bivalents, so the orientations of bivalents is independent. And we can just see that here. So we have bivalents running down the middle, and we can see that they're being separated. And the orientation corresponds to which pole each chromosome is going to go to. We can see on this one, the purple chromosome is going to the right-hand side, but in the second bivalent, it's going to the left-hand side. So this is because of orientation. So the number of possible combinations of chromosomes, so we can see here the number of possible combinations of chromosomes depending on orientation. It's dependent on how many chromosomes there are, but there is a simple formula right here. It's just 2n, and n is the number of chromosomes. So here we've got 1, 2, 3. Sorry, this is the number of bivalents. Number of bivalents. So we have 2 n's, and we have 3 bivalents. So we're going to have 2 to the power of 3, which is essentially 8. So this gives us 8 different, this will give us 8 different possible combinations of chromosomes, which we can see here. All the different possible combinations, which is a number of 8 possible combinations. Halving the chromosome number, understanding 6. So in meiosis 1 of the first division, the centromere does not divide. So it does not release the chromatids. In fact, the whole chromosomes themselves move to the poles. So if I go back to this image right here, we can see the whole chromosomes are going to the poles on either side. The sister chromatids are not being detached in meiosis 1. And this is called disjunction, so the separation of homologous chromosomes. So this right here, when they're going to opposite poles, this is called disjunction. And this halves the number of chromosomes when the cell divides in meiosis 1. Application 2, obtaining cells from a fetus. So in 3.2, we talked about a karyogram, and we talked about how we use fetal cells to get a karyotype or a karyogram. And how do we obtain these fetal cells? Well, there are 2 procedures that are used to obtain these cells. And one of these procedures is called amniocentesis. And this involves passing a needle through the mother's abdomen, which is guided by ultrasound. Well, the purpose of this is to extract amniotic fluid containing fetal cells from the amniotic sac. And we can see that amniotic sac right here. So inside the sac, we're going to have amniotic fluid, which we extract and get the fetal cells from there. There's a second procedure called coronoic villus sampling. And it essentially involves a sampling tool which enters the vagina to obtain cells from the chorion. The chorion is one of the membranes from which the placenta develops, and we can see this simple procedure right here. And this is just a comparison between the 2 different procedures, and let's compare that with data right here. So with amniocentesis, it's done later in pregnancy, but has a 1% chance of miscarriage, compared to coronoic villus sampling, where it's done early in pregnancy, but we have 2% chance of miscarriage. Skill number 1, diagrams of the stage of meiosis. So this is pretty straightforward. You just need to know how to draw the different stages for each stage of mitosis. And an important thing to know right here is because we have division occurs twice, we have the same name, so prophase, metaphase, anaphase, and telophase from mitosis, but now we notate them with Roman numerals with 1, and if I go on this side, 2. So we have prophase 1, prophase 2, metaphase 1, metaphase 2, and so on. And it's just important to memorize and learn how to draw these. And this side as well for the second division of meiosis. Meiosis and genetic variation. So every gamete is going to have a new combination of alleles, so meiosis is essentially a source of endless genetic variation. In meiosis, there are 2 processes that lead to genetic diversity. The first process is random orientation of bivalents. So this generates genetic variation among genes that are on different chromosome types. So for every additional bivalent, the amount of chromosome combinations doubles, and the possible combinations is given by the formula 2n, which we discussed earlier. The second process is crossing over. Without crossing over, combinations of alleles and chromosomes would forever be linked together. So let's say, if I get my pen, let's say if we have these 2 combinations of alleles, this is generation 1 and generation 1. And in generation 2, the organism is still only going to have this combination because there is no crossing over and you can't have variation in the combinations. Let's say crossing over was possible, and in generation 1, we're of course going to have the original combination of alleles rather, right here. And as we go to generation 2, the possible combinations come up, and these give us new combinations of alleles. So this is why crossing over is imperative for genetic diversity. Understanding 8, fertilization and genetic variation. So the fusion of gametes to produce a zygote is significant such that it is the start of the life of a new individual. So we can see that the fusion of these two gametes right here produce a zygote, and that creates a new individual or organism being born. And it allows alleles from two individuals to be combined into one individual. So the alleles from these two gametes are combined into one individual right here. And these combinations of alleles are unlikely to ever have existed before. The fusion of gametes promotes genetic variation in a species, and genetic variation is essential for evolution. So if you remember back in, look at my pen, if you remember back in 3.1, we talked about how genetics itself is split into two. And the first one we discussed was heredity. And heredity is inheriting traits. And the second one was variance. Right? So how do individuals vary from each other? And this variance part of genetics itself is linked towards these 2 points. Right? Because the fusion of gametes promotes genetic variations in a species. It also links to this point right here, because the combination of alleles are unlikely to ever have existed before. So this is going to differentiate individuals in the same species. Application 1, non-disjunction and Down syndrome. So in topic 3.2, where we talk about chromosomal abnormalities, we talked about something called a trisomy disorder. And we know this is when you have 3 copies of chromosome instead of 2. So we know that Down syndrome from topic 3.2 is something called trisomy 21. Now let's talk about non-disjunction. We know that disjunction, from talking about that earlier in this topic, is when 2 homologous chromosomes in a bivalent go to opposite poles, right? This is called disjunction. But what happens, let's say that non-disjunction occurs. What is non-disjunction? Well, essentially, non-disjunction is the failure for the homologous chromosomes to separate during anaphase. So let's say non-disjunction, both of the homologous chromosomes in our bivalent, are going to move to the same pole. So this is going to produce gametes that either have an extra chromosome or are deficient in one. And if this gamete is involved in human fertilization, the resulting individual is either going to have 45 or 47 chromosomes, because we know that normal humans, we're going to have 46. So if you have an extra, you're going to have 47, and if you're deficient in one, you're going to have 45. If we have 47 chromosomes, we know that from 3.2, we know that if you have 47 chromosomes, you're going to have something called a trisomy disorder, having 3 copies of a chromosome instead of 2. So trisomy 13 is something called Patau syndrome, and this is just having 3 copies of the 13th chromosome instead of 2. And Edwards syndrome, which is trisomy 18, is essentially just 3 copies of the 18th chromosome instead of 2. And the number after trisomy essentially just indicates the chromosome number being affected. Klinefelter syndrome is, although it is a trisomy syndrome, it's quite different from the first 2. So this Klinefelter syndrome is an abnormality on the heterosome chromosomes, right? It's an abnormality on heterosome. And this is essentially just a sex chromosome. And so we have an extra X chromosome, and this only occurs in males. So the sex notation is going to be XXY, because they have an extra X chromosome here. But let's say an individual has 45 chromosomes. What's an example of a syndrome for that? Well, an example of that syndrome is right here. It's called Turner syndrome, and this only affects females. So this is when they are deficient in the chromosome, specifically an X chromosome. So they only have 1 X chromosome. So their sex notation for the chromosome is just going to be X. The image right here shows us non-disjunction occurring. So because of non-disjunction, one cell has no chromosomes at all, and this is going to die. It's going to end up dying. And this cell right here, after it's been divided, it has two. So this is what the product of non-disjunction is. Both the chromosomes go to the same pole, and now they're in the same cell. And eventually, the zygote is going to have trisomy. And in this example, it's 3 chromosome 21, which is trisomy 13 again. Sorry, trisomy 21. Trisomy 21. And we know that this is Down syndrome. And we discussed this in 3.2. Non-disjunction and Down syndrome application 1, that concludes us with topic 3.3 meiosis.